For this project, which is supervised by Petra and by Egidio, who also joined, I think, in the meeting, we are working with 3 postdocs together, Alice, Dennis, and me, and we are putting TVB multiscale to use. So what we are trying to do is to really show use cases here. Actually, there will be 2 of them, and show how you can use all the available software in EBRAINS to answer some scientific questions. Both of the presentations that will follow now are actually work in progress. So please bear with us, but also we welcome your feedback at any point, because yeah, we are still very actively working on this and would like to hear what your thoughts are on this. Next. Yes, so why are we doing this? Why the cerebellum? So the motivation behind here is manifold. I listed 3 points here, but there are of course more. So first of all, there was a recent paper by our collaborators in Italy that showed that actually including the cerebellum in the connectome and running TVB simulations for humans was actually quite beneficial in moving closer to reality. So the matching between the empirical and the simulated functional networks was significantly improved. That was just published and made us actually think about diving more into this and also involving NEST with it. Luckily, our collaborators in Italy, they have a finished NEST scaffold model of the mouse cerebellum, which is already validated with empirical data. So that's nice, something that works in NEST already. And then we have the virtual mouse brain, of course, and the use of the mouse LM brain atlas is also great here. So all these ingredients together, we will use them now to answer the following research questions, which are more thought of as answering them in the next couple of years, but here for you to give the direction. First of all, I would like to investigate the impact of this new cerebellar spiking network on the mean field dynamics, and also vice versa, meaning actually that looking at the impact of the mean field dynamics and seeing if that can help us to actually also improve on the cerebellar spiking network. So both directions. The next point would be to embed this combined model of spiking and mean field dynamics, so the TVB NEST model, and actually run closed-loop simulations with it to investigate the data of tasks involving sensory motor tasks, where we have the data from our collaborators of some nice mice experiments. And that's the last point. It would be very interesting, of course, in the end, what's our ultimate goal, to bring this back to the human and to humanize kind of our results and say, okay, the brain diseases are what we are investigating, what's our focus on, and can we actually, with our new model that we build with the cerebellum and with the rest of the brain, investigate what is altered in different brain diseases, because the cerebellum plays a role actually in many of those brain diseases. So it would be great to have a more accurate model to investigate changes there. Okay, next. So this is a broad overview how our multiscale model that we built so far looks like. We base it on the Allen Brain Institute data of the mouse connectome. And then we actually have the cortex just modeled by TVB, or here TVMB, so the virtual mouse brain. And for now, using reduced Wang-Wang models. We will see how far we can get with that. And as the cerebellum itself, we will model with the NEST model from Casali et al., so from our collaborators. And there is a lot of detail involved and a lot of cellular different properties that we can actually give parameters there and fix there. And Alice will continue and give you an overview of that.
So the model we are using in this first TVB NEST use case with the cerebellum includes, of course, the cerebellar circuit and the main regions of the brain projecting to and from the cerebellum. As we can see in this simplified block diagram here, the cerebellum receives input from sensory pathway areas through the mossy fibers, which then project both directly to the output of the cerebellum, which is the deep cerebellar nuclei, and also to the granular layer input stage of the cerebellum, which here is in the granule cells and Golgi cells, and then goes back to the rest of the cerebellar circuit up to the Purkinje cells, which in turn inhibit the deep cerebellar nuclei cerebellar output. Another input to the cerebellum comes, on the other hand, from the inferior olive nuclei, which directly project to the Purkinje cells. So within the cerebellar circuit, we have two levels of integration. At the Purkinje cells level, integrating the MOSI fiber processing throughout the different layers of the cerebellum, and the direct input from the inferior olive. And finally, the integration at the level of the deep cerebellar nuclei, which integrates both the direct MOSI fiber input and the Purkinje cell inhibition. The result, the output, is a projection to the motor areas, giving the cerebellar contribution to motor commands. The starting point of our study is a cerebellar scaffold model, which we are developing at the University of Pavia, and you can find all the code in the links posted in the slide. The cerebellar scaffold model is aimed to realistically reproduce the cerebellar network. So the first stage, which is the reconstruction, includes the first phase of placement of neurons, starting from density data of the different neural populations and from dimension of the different cells in the circuit. And once the neurons have been placed in the circuit, they are connected based on the overlap of their morphology, because each neural population can be associated to a morphology. So from the overlap of them with some tolerance, we can identify the connections. The result is that the model has a data-driven topology, which reproduces the quasi-crystalline structure of the cerebellar circuit. Once we have reconstructed the circuit, we can simulate at different levels of detail. The one we are interested here is at the level of spiking neural network, which we did using the NEST simulator. Also, in this case, we wanted to stay close to data, so the different parameters in the model were tuned based on experimental data. Specifically, for example, the single neurons were modeled as point extended generalized leak integrated fire neurons, with parameters optimized to reproduce the different electroresponsive patterns of the neural populations in the circuit. Then also the synapses, so the weight and delays of the connections in NEST were tuned in order to reach in vivo population firing rates. And finally, the stimuli provided to the network reproduced the spiking patterns of sensory-like stimuli. So the result is that also the network dynamics is based on data. What we did starting from this point, so from a network which is optimized at baseline, in baseline conditions, to validate if it was able also to reproduce realistic responses to specific stimuli, which in this case are associated to a typical cerebellum-driven task, which is deadly classical conditioning. These stimuli are given to the cerebellar network considered as a standalone block. So the interface to the external brain is modeled as spiking patterns, and the input are encoded as spiking patterns to the MOSI fibers, as you can see in the left, as a constant spike in Poisson process, and to the inferior olive as a burst to simulate the two types of stimuli involved in the task. The output of the cerebellum is then decoded from the deep cerebellar nuclei spiking pattern. These two pathways are causing different responses in the cerebellar network, which we were able to reproduce. Specifically, the MOSI fiber pathway is causing an overall increase of the population firing rate in the whole network with a balance of excitation and inhibition, while the bursting input is causing nonlinear responses in the Purkinje cells and in the deep cerebellar nuclei. So the network was also able to reproduce the challenge of simulating stimulus response patterns of different nature. The next step, which is the objective of this first proof of concept use case, is to put the standalone spiky neural network validated model within the rest of the brain into a TVB nest framework. We focused for this first example on the first pathway, so the one through the MOSI fibers. And as Jil said before, we use the connectome in the TVB mouse, also based on Allen Brain Atlas data. And for this example, we used only the regions with a volume higher than 2 millimeters cubic. And we reduced the cerebellum from 33 regions to just one region. Focusing on the pathway from the MOSI fibers, according to the connectome we obtained, the cerebellum is receiving input from the isocortex and also from subcortical areas like the medulla in the brainstem, the midbrain, and the pons. To simulate the stimulation pattern through the MOSI fibers that we have seen before, we decided to stimulate the intermediate reticular nucleus in the medulla, which projects directly to the MOSI fibers and thus to the input of the cerebellum, but also indirectly through the inferior colliculus in the midbrain and through the pontine nuclei in the pons. On the other hand, the output of the cerebellar spiky neural network collected at the deep cerebellar nuclei population is then conveyed to a TVB node representing the motor cortex, again based on the connectivity from the TVB connectome. The interface between these two nodes has been specifically modified for this use case, as Dennis is going now to explain you more in detail.
Right. So, Alice, I will ask you to change the slides when it's necessary, okay?
Yes. Okay.
Yeah. So, turning into the implementation in TVB Nest, we first had to choose a TVB model. And since for the moment we're looking into linear responses in TVB, we made a very simple linear model where for most of the nodes that are implemented in TVB, the activity is just, it just depends on the long range coupling that receives from the other regions and from a possible external stimulus, which is 0 for all regions except for the one we stimulate for some time, the right medulla. And for the regions that are modeled in Nest, the rate is given by following the input we get from Nest, as you're going to see in the next slides. So this was only one region for the moment, since we merged cerebellum to be one region only. And also, as you saw in the previous slides, we get the activity from the DCN cells. So yes, so for the interface from TVB to Nest, it is a kind of coupling we implement by using the concept of a TVB proxy node inside the Nest network. This is a device that can generate spikes and mirrors the activity of the corresponding TVB node the signal is coming from. So we take the instantaneous rate at its TVB time step to update this device inside Nest. We multiply with a constant that could represent the number of neurons that are actually in this population, and we send the spikes towards the target population at the target region. In this case, it's only the MOSI fibers inside the cerebellum. Let's go to the next slide, please. For the other way around, it's a kind of update, as you saw. It concerns only the cerebellum node that is modeled in Nest. And all we need to do is measure the spikes, count the spikes of the DCN population using a spike detector device. And at every time step, divide by the total number of neurons and by the length of the time step to get an instantaneous spike rate, which we sent to TVB to be integrated, as you saw, in order to smooth it out a little bit. So this is how at its TVB time step, we update the cerebellum state from Nest. And this means that cerebellum is keeps being represented inside the TVB connectome. There is no hole in the brain. We keep the integrity. We maintain the integrity of the TVB connectome, so everything else, couplings, history, or neuroimaging monitors can work as usually. Yep. Next slide, please. So I'm only showing you a little bit of the API for the user who doesn't really need to know too many things about TVB or Nest in order to configure such an interface. So for the TVB to Nest coupling, all we need to do is to define a dictionary of properties, a Python dictionary of properties like this. In the future, it could be a JSON file or some other configuration file, where we define the model of the device we need to use as a proxy node, some parameters. The important thing is to weight the connections from that proxy TVB node to the target Nest nodes, taking into consideration the connectome of TVB and the delays as well. And yeah, this is the most crucial part, is where we define the exact connections. So we say take the activity of the specific TVB state variable, the rate, and send it to the specific population in Nest. And the final entries, source nodes and target nodes, are used in case we want to differentiate this interface between different pairs of nodes. In this case, it was all TVB nodes against one Cerebellum node, so we didn't have to say anything there. And similarly, for the other direction of the coupling, we need to use a spike detector device. We might wait a little bit, scale the input, and mainly define the connections which go from the DCN cells of Cerebellum to update the input rate we have created as a state variable in the model. So that's all. With a few lines, we can configure this interface. So our first step was to validate our model against the code we got from Alice. So this is still only Nest simulation, but here on the right, we built the whole network using our interface with in a similar fashion with configurations. And also we have compiled the module we got for the neurons, the new Nest models we got from Alice. All this we did through our interface, and for the moment we don't want to focus on the details of the figures. Only visually we can see that we get the same results, so we are sure that the Nest network is the same. And then on the next slide, yeah, and we had to do several modifications to allow for the specific scaffold connectivity to be implemented through our interface. Yeah, so here now we already connected TVB to the Nest network, and we have some first results. I will start showing you. On the right is the time series we get from TVB. On the left is the activity of the Nest Cerebellum. And for the moment, I will only explain the orange line. I'm not sure what color you see there. Give me a second. Yeah, it should be an orange line, which is actually the activity of the region we stimulate, the right medulla, and you can see the effect of the stimulus, just a linear response. And now Alicia again will explain you the details of how this activity propagates through the cerebellum. And when we come back to TVB, I'll also explain the response to TVB.
Yeah, so the effect of the stimulus on the medulla throughout the two direct and indirect pathways we saw before in the connectome is causing the expected firing rates in the mossy fibers that you see in the left, both in background conditions with low around 4 Hz firing rate and during the stimulus. Then from the mossy fibers, this stimulus propagates throughout the spiky neural network, both directly from the mossy fibers to the deep cerebellar nuclei, which is the last block on the right, as you see with the excitatory connection arrow, and indirectly through the Purkinje cell inhibition. So the output of the cerebellum an overall increase of the deep cerebellar nuclei firing rate during the stimulus is the result of the balance between the excitation directly from the mossy fibers and the inhibition from the Purkinje cells, and it's at rates expected for this kind of stimulation. Then from the deep cerebellar nuclei spikes within the NEST network, the signal goes back to TVB, and Dennis will...
Yes, so on the figure, thanks, in the figure at the bottom, you can see the spiky input from the DCN cells to the cerebellum node of TVB. And through the linear integration you saw, we can see this curve, the blue curve that is formed on top. I forgot to say that you can see a little bit of the indirect path activity the stimulus invokes in the pons regions, but it's relatively low. Instead, you cannot really see for the moment any linear response to the cerebellum activity. The reason for this is that in this connectome we have the cerebellum is weakly connected to the other TVB regions. So it cannot really cause for the moment any linear response. This is an issue to deal with in the future. In general, how to invoke activity related to a task to TVB, because there is no plasticity for the moment. So yeah, this is under working process.
All right. And then just to give you a short summary of what we just presented to you, in case also you arrived later after lunch, we were actually able to integrate the NEST cerebellum model in the TVB NEST framework. And this NEST cerebellum scaffold model actually includes specific neuron features. So you can, on a cellular level, adjust things, and then actually you can see the effects on the TVB mean field dynamics. And as a second point, we could tune this implemented model already such that we were able to validate the only NEST part of our implementation with the only NEST part from Alice, so from our collaborators. And we could also validate this already when including the TVB part as well. So that seems to provide some realistic firing behaviors that were expected based on the biological pathways. So in total, we built a TVB NEST model, including the cellular model of the cerebellum, with realistic firing behavior. And just to give you an outlook on some future work, there we have, of course, a lot of work to do. It's all work in progress. Most of the results that you saw were generated very recently and yeah, are still kind of very fresh for us. And but just to give you some long-term goals, we put some points here to think about. First of all, as was mentioned, so far we summarized the whole cerebellum into one region. And of course, there is much more to the cerebellum than one region. Actually, we have up to 33 different cerebellar regions that we are aiming for to actually give them all different parameters, fit them in the most realistic way to get this model as close to reality as possible for the mouse. And then we have, luckily, a lot of data of experiments available from mice experiments. So we want to actually compare that and see if we can actually model stimuli responses or responses to motor or behavioral tasks of mice. Along the way, of course, this point, we have to be very careful because there we have to actually improve the task responses that TVB is providing. So far, there's not a lot of implementation on task behavior in TVB because there's a lot of transient behavior, which makes it more difficult to capture those specific phases of the simulation. But actually, yeah, that's something along the way that we put on our list to improve on. And at the third point, we want to extend it to other regions of the mouse brain and also human brain, which are very interesting, like the thalamus or the hippocampus or the basal ganglia. And actually, that will be the next talk. And as a last point, always in mind when simulating the mouse to translate it back to the human, use human tracts, actually DTI data, and do the same thing as in a TVB NEST environment. So just to thank all the people involved. Next slide. Yes. of course, the mice, but also the supervisors, Petra and Egidio in Italy, and all the collaborators from Italy as well, and also the other project partners from the Rising Net SGH3 project. And we have some as a last point, some references for you, where it actually includes also the bottom link is to the notebook, which we are currently working on, and which it's really accessible. You can use it to generate and regenerate the results we just presented.